Hello everyone, Mr. Linder here. Let's talk about the sliding filament mechanism, or also called the contractile cycle. So when we look at a contraction cycle in skeletal muscle, the things we really want to consider are binding, the power stroke, the release step, and the resetting step. So if you can remember binding, power stroke, release, and reset, you essentially can learn the sliding filament mechanism. So what's going on in the binding step of the contractile cycle? Well, when you look at resting muscle, the myosin head is in a cocked position with potential energy. So if we look at this position right here, this is the potential energy position. And when the myosin head is in the potential energy position, it has ADP and inorganic phosphate attached to the myosin head. In this position, you can have weak actin and myosin cross bridges forming, but the power stroke won't take place. And the reason the power stroke won't take place is because there's actually a blocking protein preventing the myosin heads, so preventing the myosin from creating a high force bond with the actin in order to actually have a power stroke. So you can have a weak interaction, so we can have a weak interaction between the myosin and the actin, but as long as tropomyosin is in the way, you're not going to have the power stroke that takes place. So in order to have a power stroke, there has to be a calcium signal. That calcium signal activates troponin, which is going to move tropomyosin. So there's another protein called troponin. So we'll label that troponin. And this, of course, was the blocking protein. So we'll label it tropomyosin. And troponin can actually move tropomyosin if calcium binds to it. So if we get a calcium ion that can bind to troponin, you can move tropomyosin out of the way. And if tropomyosin is able to move out of the way, then you can have high force bonding between the myosin heads and the actin. So calcium is important to allow for the high force binding to take place between myosin and actin. So that's the first part, the binding part. Once tropomyosin is moved and the myosin head attaches to the actin protein, then you can move on to the power stroke step. So if we look at this new figure here, really what we'll have is we'll have tropomyosin moved out of the way. Here's our troponin protein, and we can have troponin in several locations, and calcium is attached to the troponin. So we'll put some calcium attached to the troponin. And when calcium is attached to troponin, we've moved the tropomyosin out of the way. And so now we have the myosin head bound to the actin in a strong binding position. So instead of a weak position, now we have a strong, high force binding position. When you're in the high force binding position, you then can release energy. So we can release that inorganic phosphate, and that leads to the power stroke that takes place. And basically the power stroke is just the moving of the myosin head. So the myosin head moves forward, and when it moves forward, that means that the actin protein is able to slide towards the middle of the sarcomere. So it's moving towards the M line. So the release of inorganic phosphate gives us the power stroke. So in order for myosin to bind to actin, we used calcium. In order to initiate the power stroke, we release inorganic phosphate so that that myosin head can bend, and that gives the power to slide the actin over the myosin. So it's sliding towards the M line and it's making the sarcomere shorten. In order for myosin to detach from the actin, there needs to be another molecule that can separate the proteins. If you don't have another molecule to separate the proteins, you're going to stay stuck in a state of rigor. So ADP can come off of the myosin head, but without another molecule, these proteins will stay attached to each other. So the myosin will stay attached to the actin in what's called the rigor state. So myosin releases ADP at the end of the power stroke, but the myosin and actin are in a state of rigor. They're basically stuck in a contractile state unless another molecule comes along. That other molecule that's going to release myosin from the actin is ATP. So we need ATP to bind to the myosin so that it will release from the actin. So the release step is ATP attaching to myosin, causing the myosin to release from the actin. Once myosin has released from the actin, we need to hydrolyze the ATP so that we can put the myosin head back into its potential energy position. So this is the reset step. So what's going to happen is ATP is going to get hydrolyzed into ADP plus the inorganic phosphate. And we're going to be able to take that myosin head and reset the myosin head back to the original position. So we come down to the beginning part of the diagram again, and now we have the myosin head back in its ready position with the ADP and the inorganic phosphate in that potential energy state, ready to go through the process of the contractile cycle again. So that ATP hydrolysis resets the myosin head, making it available to be used again. So if we review binding, power stroke, release, reset is the contractile cycle. In order for binding to occur, we need calcium. In order for the power stroke to occur, there needs to be a release of energy, inorganic phosphate. In order to release the myosin from the actin, you need ATP. And then in order to reset the myosin head, you need to hydrolyze the ATP back to ADP plus inorganic phosphate. And so that takes us through the entire contractile cycle. From a clinical standpoint, if you don't produce enough ATP, that would prevent the myosin from detaching from the actin. So a good example of this is after death, when the body stops producing ATP and the supplies of ATP are exhausted, muscles are going to enter a state of rigor mortis. Rigor is that contractile state. Mortis is referring to after death. So it's a state of muscle contraction after death. Basically, the myosin has attached to the actin because there's been a release of calcium from the sarcoplasmic reticulums. The binding occurs and then the power stroke occurs. But once the power stroke occurs, you're going to be stuck in a state of rigor when you can no longer have the production of ATP. So when cells are dead, they're no longer performing metabolism to produce ATP, and so there's no ATP to release the myosin from the actin. Rigor mortis typically can last anywhere from 24 to 48 hours after death until the cells are going to eventually begin to break down, and there's going to be muscle protein decay, and then eventually the rigor state will be broken, and then the musculature can go back to a flaccid state. So that is the contractile cycle, or the sliding filament theory. I hope that helps. Take care.